Hello everyone, this is Vaibhav Shandelia, MBBS student from ESIC Government Medical College, Hyderabad. And today we're going to talk about a very important microorganism, probably one of the most, which is the HIV, human immunodeficiency virus. So it is the part of the family lentivirus, and it is of two main types, HIV-1 and HIV-2. And out of this, HIV-1 is the more pathogenic and more important form to us. So let's start. This video will be in the form of a table more than a mind map, which I found more effective. The subheadings are structure, serotyping, pathogenesis, lab diagnosis, and Naco strategy. So let's start with structure. So it has three main elements, which is the envelope, nucleocapsid, and genes. So coming to the envelope is made up of obviously two parts, the lipid part and the protein part. It is same for all the viruses mostly. So lipid part and protein part. Lipid part is derived from the host cell, while the protein part has two important glycoproteins, which are GP120 and GP41. GP120 is responsible for the spikes on the surface of HIV, while GP41 is responsible for the transmembrane adhesion pedicles. Basically, it helps in fusion. I'll mention this later. Again, so GP120 responsible for spikes and GP41 responsible for the transmembrane adhesion pedicles. This can be seen in the diagram. This is the outer is the envelope, and this envelope has a lipid portion, and it also has a protein portion. The protein involves this, which is the GP120 spike, and this, which is the transmembrane adhesion pedicle, or the GP made of the GP41 protein. So this is the lipid and the protein making the envelope. Next, we have the nucleocapsid. The nucleocapsid has basically makes the inner core, and this contains of two identical single-stranded positive sense RNA. Very important. Two identical single-stranded positive sense RNA. Very important. And also it has three very important enzymes: integrase, reverse transcriptase, protease. Integrase, reverse transcriptase, protease. You can see this here. These are the two single-stranded positive sense RNA. We have reverse transcriptase, integrase, and also there is protease inside. So these are the three enzymes and one RNA inside the nucleocapsid. Okay, this inside is the core, the nucleocapsid core. Okay, so now the third element, the genes. Here there are three main genes: the GAG gene, Pol gene, and ENV gene. GAG, Pol, and ENV. These are the three genes. GAG gene codes for the outer shell plus inner core. Okay, the outer shell and the inner core. So basically we have the P55 protein, which is cleaved into P18 and P24 plus P15. You have to remember this, write this one or two times and you'll easily be able to remember this. P55 cleaves into P18, P24 and P15. P18 is responsible for the shell, while P24 plus P15 is responsible for the core. This will be, re-emphasized later because P24 is a very important antigen. So P55 protein is broken down into P18, 24 and 15. Coming to the Pol gene is responsible for formation of the enzymes. Which enzymes? As I mentioned earlier, reverse transcriptase, protease, and integrase. Okay, while the N gene is the main one. It's the main gene, and which is responsible for, as the name suggests, of envelope. So here we know that GP120 and GP41 are the protein components of the envelope. So what are these made up of? GP160. Almost 120 plus 41 is equal to 160. So almost. So you can remember it that way. GP160 cleaves into GP120 and GP41. And this GP120 attaches to the CD4 T cells, and GP41 is responsible for the fusion. So basically the spikes of GP120, they attach to the CD4 receptors, receptors on the CD4 cell, while the GP41 is responsible for the fusion. This will be explained in detail further. Now all vaccines against the HIV are formed against the ENV genes. But at the same time, these ENV genes are the most mutated genes. Hence, HIV always tends to escape the host immune system, and so HIV is a very dangerous disease. Okay, so it's obvious. All the vaccines against ENV genes which are most mutated. So basically what did we learn in structure? There is an envelope made up of a lipid and protein part. Protein part is made up of GP120 and 41. GP120 is the spike, while GP41 is the transmembrane protein. Nucleocapsid is the inner core, which consists of two single-stranded positive sense RNA and three enzymes: integrase, reverse transcriptase, and protease. Okay. Now there are also other accessory proteins like VPR, etc., which I will mention later. But remember, there are other accessory proteins as well. Then there are genes GAG, Pol, N. GAG is responsible for formation of the outer shell and inner core made up of P55 cleaving into P18, 24 and 15. Then Pol responsible for the enzymes, and envelope gene responsible for GP160, 120 and 41. Coming to the serotyping, now as I said, HIV1 and HIV2 are the two forms. Now we'll talk about HIV1 mainly. So HIV1 has three serotypes, M, N and O. M, N and O. Now out of this, M stands for main, so this is the main subtype, and this main subtype, main serotype further has subtypes which are A to K. So M has further subtypes A to K. Clear? And these A to K subtypes further have subtypes. Okay, it's not that confusing, just read it again and again. So the A to K subtypes have subtypes which are basically A to K will have A1, A2, B1, B2 and so on, K1, K2 and like that. So these differ in basically geographical distribution and transmission, not really in the manifestations as such. But basically all you need to know is the serotyping. HIV1 is divided into MNO serotype, and M is further divided into A to K subtype, which is further divided into A1, A2, B1, B2 and so on subtype. And together, they, in different combinations, they basically form the circulating recombinant factors in different combinations known as the CRF. For example, if the circulating recombinant factor 01, which is the type of the virus, and AE means the A and E subtypes coming together in a recombinant fashion will form CRF01 underscore AE. So this is just a type of naming. Okay. Coming to the modes of transmissions, we have four main modes of transmission, which are the blood transfusion, sexual contact, the mother to baby or vertical transmission, and percutaneous injury. Okay, so basically coming to blood transfusion, this is the least common occurrence because we obviously screen these blood packets through many levels of safety, but there's also maximum risk of transmission. That means if the blood is transfused with HIV, the max chances of transmission is with this. Next comes sexual contact. Most commonly occurring form is sexual contact and maximum transmission is by anal route. 75% occurrence. Mother to baby is a 20 to 40% transmission rate. And then with percutaneous injury, we involve the needle stick injuries, tattoos, razors, IV drug abusers and all those. Now, maximum viral load will be found in the blood, CSF and genital secretions, but there will be zero viral load in the urine. You cannot detect HIV in the urine. So basically, what did we discuss? Modes of transmission, blood transfusion, sexual contact, mother to baby, percutaneous injury. Okay, good. Coming to the this was part of the pathogenesis. Coming to the infection, how it is spread, the real pathogenesis, the cycle. So basically first the GP120. See, this was the GP120 spike. So first of all, this GP120 will bind on the host cell. How? GP120 bind on the CD4, which are the basically macrophages, dendritic cells, astrocytes. And there are basically two responses which have to occur for HIV infection to happen. The first main receptor response is GP120 binding to CD4. Then the second response is GP120 binding to the chemokines. Am I clear? GP120 binding to CD4, then GP120 binding to chemokines. Which chemokines? We have CXCR4 and CCR5. Remember, pathology, second chapter, inflammation. This is mentioned. Chemokine types, CXCR4 and CCR5. So GP120 binds to chemokines and CXCR4 present on T cells and CCR5 on macrophages. Mainly, you just have to remember GP120 will bind to CD4 and also chemokines together with this main response and this co-response. Internalization will occur. So both responses occurring will help in internalization. Internalization will occur with the help of which protein? Yes, it will be the fusion protein GP41. Fusion protein GP141. So both the responses occur. GP41 helps in fusion and internalization. Now which part is internalized? The whole virus is never internalized. It is the nucleocapsid that enters the host cell. It is the nucleocapsid that enters the host cell and it releases whatever is inside the nucleocapsid in the core. Remember, what did we have? We have two single-stranded positive sense RNA, integrase, protease, reverse transcriptase. So this, along with accessory proteins and all, are released in the host cell. Now there, the single-stranded RNA with the help of reverse transcriptase enzyme will convert into single stranded DNA. And then by replication, duplication, it will form double stranded DNA. So you know how we converted into double stranded DNA. Now, what is left? The double stranded DNA, the integrase, the GAG protein, and at the same time an accessory VPR protein. Remember I mentioned accessory proteins? This is the VPR protein. So the dsDNA which was formed, the integrase which came on the infection, the GAG protein and the VPR protein, together they form the pre-integration complex. The pre-integration complex. And now this pre-integration complex, with the help of this integrase, will get incorporated into the host chromosome. Okay, so the pre-integration complex, which consists of the dsDNA, with the help of integrase, this dsDNA gets incorporated into the host chromosome, and hence now infection has occurred and there has been a chromosome mutation, and now the virus will go latent for almost a few months to 30 years as well. The average is 10 years. Clear? What did we see? We saw two responses, GP120 binding to CD4. Second response, GP120 binding to chemokines. Together, internalization with the help of GP41 protein, fusion protein. Then only the nucleocapsid enters, releases certain RNA, integrase, protease, reverse transcriptase. Then there is formation of pre-integration complex. With the help of integrase, the dsDNA in the pre-integration complex gets incorporated in the host chromosome, goes latent for months to years. This was the pathogenesis part 1. Next pathogenesis is the disease progression. Call this the manifestation, call this the complication. The disease progression is in the form of 4 main headings. First, there is acute HIV. Then there is asymptomatic stage. Then there is persistent generalized lymphadenopathy. Then symptomatic HIV, and then AIDS finally. Okay, so what did we have? First we had an acute HIV, then we had an asymptomatic stage. Then after asymptomatic stage, the infection started to come back. So we had persistent generalized lymphadenopathy. After that we had symptomatic HIV, and then we had AIDS. These are the stages. These are the 5 stages. Okay, the persistent generalized lymphadenopathy is a minor stage. There are 4 main stages. Nonetheless, let's start. First, acute HIV. So after infection, after the whole pathogenesis mentioned over here, after that the HIV is carried to the lymph nodes. There it replicates in the T cells. Replication. So infection, then carried to the lymph nodes, replication. Initially it will destroy the T cell. Initially this is a new infection. Body does not have any antibodies, any host in response, nothing is there. So initially it will destroy the T cells and spill in blood. Basically what we witness is primary viremia. When it just enters, primary viremia, and there is a significant drop in CD4 to be noted. This is the acute HIV. But then the asymptomatic stage kicks in. How? Because now the cell-mediated immunity, the humoral-mediated immunity, all the CMI and humoral immunity, both bring back the CD4. They fight with the virus, they decrease the viremia and bring back the CD4 to normal levels. After that, because the levels came back to normal, the virus goes to latency for 10 years. It is not able to fight. After 10 years, when it has to come back from latency, we will see a stage of persistent generalized lymphadenopathy. That is lymphadenopathy persistently for more than 3 months. That means the virus is coming out of latency. Next, after that is completely left latency, we'll see the symptomatic HIV, wherein the viremia will rise again like it did earlier. But this time the virus is much stronger. So the CD4 cells will drop extremely less CD4 cells. What will we see? Diarrhea, weight loss, nausea, vomiting, anorexia, fatigue, malaise, and opportunistic infections. All these general or severe features. Okay? Diarrhea, weight loss, nausea, vomit, anorexia, fatigue, malaise, opportunistic infections. Finally, AIDS occurs. There is a rapid fall in CD4. Less than 200 cells per microliter. Less than 200 cells. Remember this value. High viral load. Viremia has increased too much. Lymph nodes are destroyed. The infiltrated lymph nodes are destroyed completely. Opportunistic infections. Remember the name of few opportunistic infections? Pseudomonas, pneumocystis jirovecii, Kaposi sarcoma, cytomegalovirus, HSV, HIV encephalitis. Opportunistic infections that occur. So basically what did we see? First, acute HIV. HIV starts, infects, causes primary viremia. There's a drop in CD4, but then asymptomatic stage. CMI and humoral immunity bring back the CD4 count, decrease the viremia and latency. Then comes out of latency when there is persistent lymphadenopathy for more than 3 months. Symptomatic HIV. Viremia occurs again and we see all these symptoms. And then finally AIDS occurs when the CD4 counts decreases below 200. There is high viral load, lymph node destroyed and opportunistic infections. Okay. Coming finally to the lab diagnosis, also important. Remember the 3 C's. The 3 C's, very important. Consent, confidentiality, counseling. You will write all this in all of your answers. You have to remember this: consent, confidentiality, and counseling. 3 C's. Let's start. Antibody detection first of all. How? ELISA, obviously. Antibody detection by ELISA. And then now classical rapid or simple tests are done. Rapid or simple tests. Which are these? You need to remember this: dot blot, immunochromatography test, latex agglutination, and dipstick. Dot blot, ICT, latex agglutination, dipstick. These are the rapid tests for quick results. Then Western blot. This is the most commonly done. Western blot most commonly done. But what happens in Western blot? Basically, Western blot is for detection of antigen and antibodies. Basically, it detects various antibodies against the separate antigens. Antibodies against the antigens is what is detected in Western blot. For example, antibody against the GAG detected, against POL, against N gene. So antibodies are detected in Western blot. Now, according to the WHO, if more than 2 envelope components antibodies against are positive, antibodies against 2 envelope components are positive, then we are HIV positive. You know which components? GP160, 120 and 41. If any 2 out of these 3 are having antibodies in the serum, that means WHO says it's positive, HIV positive. Clear? So antibody detection. Then next we have detection of P24 core antigen. Remember the P24 core antigen. Remember P55 splitting into P18 and P24 plus P15. Remember? And then this was forming the shell and this was forming the core. This is exactly that P24 core antigen which is responsible for, remember this, confirmation of HIV AIDS. Very important. Diagnosis of HIV in the window period. That is from infection till formation of antibodies in the window period. Then it helps in monitoring of progression and its diagnosis of HIV in infants. Helps in diagnosis of HIV in infants. This is the P24 core antigen. Very important, window period. Next, finally, we have viral RNA detection. Viral RNA detection is the gold standard method. Gold standard method for detection. Viral RNA detection. Single stranded. Remember 2 single stranded positive sense RNA. We detect that via the reverse transcriptase PCR, NASBA, which is basically the nucleic acid sequence based amplifications. Nucleic acid sequence based amplifications, and real-time PCR, and detection of drug resistance is also done via this process only, if there is any drug resistance. Then finally some non-specific tests, which is the CD4 count or beta-2 microglobulin detection. That's it. Okay, so what were the things we said in the lab diagnosis? First 3 C's. Then in antibody detection, ELISA, rapid test, dot blot, ICT, latex agglutination, dipstick. Western blot most commonly done. Basically, WHO says any 2 proteins antibodies should be positive against the envelope genes, which 160, 120 and 41. Then P24 core antigen. Remember, it is responsible for window period diagnosis in window period and in infants, helps in progression monitoring and confirmation of HIV/AIDS. Then viral RNA detection is the gold standard done by PCR, NASBA, and it also detects any drug resistance. Non-specific test, CD4 count, beta-2 microglobulin. That's it. Finally, let's talk about the Naco strategy, National AIDS Control Organization strategy. So this is a very important strategy. There are 4 strategies named: strategy 1, strategy 2A, strategy 2B, and strategy 3. Okay, and these are employed in 4 different conditions. First of all, let's talk about strategy or algorithm 1. This is for transfusion transplantation safety. You get a blood bag. How will you'll basically this is how you'll test for HIV. You get a blood bag. You'll test for HIV like this. How? You only need 1 test kit for HIV is required. If you get HIV positive, it's HIV. If the blood contains HIV virus, HIV positive. If it's negative, considered negative. You discard the blood, destroy the blood if it's destroy the blood if it's positive, and if it's negative, you use it. Next, strategy 2A. This is the strategy for surveillance. In general population, you use it without any consent, just for surveillance. 2 test kits are required. If you get negative report, then you just report negative. If you get positive, test kit reports positive, then you have another test kit. If this test kit also reports negative, then only you report negative. 1 positive, but then 1 negative, you report negative. If it comes positive, there are 2 consecutive positives, so you report positive. You just have to make this on your own on a piece of paper and you'll remember this easily. So, for surveillance without consent, 2 test kits. If it's negative, report negative. Positive, do another test. Again positive, then report positive. If negative, report negative. Then third, this is for diagnosis of an individual with AIDS and which is showing symptoms. Okay, showing symptoms of AIDS, diagnosis of individual with for the diagnosis, you need 2 or 3 test kits depending on the condition. If it's negative, basically just report negative. If it's positive, do another test kit. If this is also positive, 2 consecutive positives, you report positive, and then you go for post-test counseling. If you get first positive, but you get second negative, and then you get consecutive third negative also, 2 consecutive negatives, you report negative. But if you get this third one positive, indeterminate. You cannot determine if it's HIV positive or negative. Do the test again after some time. Now, fourth strategy, or the strategy 3, basically, this is to detect HIV infection in asymptomatic individuals. HIV infection in asymptomatic individuals. Now, you need 3 test kits. If you get negative, obviously again report negative. If you get positive, another test. Now, here it's interesting. If you get negative, first you got a positive, then you got a negative, then you do third test. If again you got a positive, indeterminate. You can't determine. If you got a negative, then technically, you got 2 negatives. But if the person is from a high risk background, it's considered indeterminate. If a person is from low risk background, it's considered negative. Remember? So, 1 positive, then second test, that comes negative. Then again, if that comes negative, there are 2 consecutive negatives, then you do this. If this test comes positive, then indeterminate. Now, first test comes positive, second test also comes positive, third test also comes positive. Obviously, that's a positive, and go for post-test counseling. If this comes negative, you cannot say for sure, indeterminate. Okay? A little confusing, but it's not that hard, to be honest. Just write it 2 or 3 times and you'll remember it. So, we have 4 strategies: 1, 2A, 2B, and 3. First was for blood transfusion, second was for surveillance without consent, third was for patients who come with symptoms of AIDS, and fourth is for HIV infection in asymptomatic individuals. This is it for HIV. Let's recap it. Structure: we had envelope, nucleocapsid, and genes. Envelope had lipid protein part, GP12041. Nucleocapsid had structures like 2 identical single-stranded positive sense RNA, enzymes, 3 integrase reverse transcriptase protease. Then genes, we have GAG, POL, ENV. GAG was responsible for P55 splitting into P18, P24 plus 15, POL for the enzymes, and N for GP160 splitting into 120 and 41. Serotyping, we had, remember, HIV MNO subtypes, then A2K subtypes, MNO serotype, A2K subtypes, and then subtypes. Then modes of transmission was blood transfusion, sexual contact, vertical transmission, and percutaneous injury. Finally, infection was how 2 responses were needed: GP120 binding to CD4 and GP120 binding to chemokines. Then internalization with the help of GP41. Nucleocapsid enters. Then there is reverse transcriptase, helps the single-stranded RNA convert into double-stranded DNA. Then formation of a pre-integration complex with the help of integrase incorporated in host chromosome. Then what were the stages? Acute HIV, you get little symptoms, primary viremia, less CD4 count. Then asymptomatic stage, you compensate, virus goes for latency. Then persistent generalized lymphadenopathy for more than 3 months. Then symptomatic HIV, the virus leaves latency, viremia increases, CD4 decreases. Then finally AIDS, when the CD4 count goes below 200 and you get all sorts of opportunistic infections and high viral load. Then lab diagnosis, we talked about 3 C's: antibody detection, ELISA, rapid test, Western blot, P24 core antigen, viral RNA detection, and non-specific tests. And then finally Naco strategy. So this was the video on HIV. I know this was a long one, but this is also a very, very, very important virus. You will definitely encounter it in many times, not just once or twice, many times. So be prepared, practice this, and all the best. Thank you.